Good afternoon, everyone. We come from a small university in 200 kilometers south to Brisbane. And some of the audience actually before I start asked me where is Southern Cross University. Is that in Australia? Yes, it is in Australia. If anyone of you have been to Byron Bay, we are probably around 30-40 kilometers west to Byron. So this is a corner of our chemistry lab, and we also have a genetic lab there as well. We got probably about 10 HPLCs, lots of LCMS, GCMS, GC there as well. And there's a TGA certified laboratory. So we have probably 500, 600 customers every year go to us. And we have easily can run around 10,000 samples with certificate through the lab. But basically, I don't do this job, and I'm academics, but we use the lab here as well to do whatever things we want to do. So today I would like to talk about a little bit sort of my hobby research. What I have done in the past couple of years is about researching about the quality of rice. So there's a particular quality of the rice I'm interested in is actually related to the type 2 diabetes. And lots of you probably heard of the story about eating white rice. You can, raise your blood sugar, and you can get type 2 diabetes. Is that true? I mean, from the sort of, if you actually go through all the clinical trial data, someone actually went through this and did what they call a meta-analysis, and then they produced this called a forest plot, which is they basically collect all the clinical trial data together and try to see when people actually did clinical trial on, their subjects eating white rice, whether they follow it up after a number of years, see whether they develop type 2 diabetes. It's quite interesting what they found is in Western populations, they have very low incidence, but in Asian population, you certainly can see the incidence is a bit high. So they get sort of give you an indication because Asian people actually tend to eat more rice than Western people. So that's why there are more people actually develop type 2 diabetes from there. So they also actually collect all the, these how much white rice they eat. So you can see here is a 0, here is a 1,000 grams per day. And it's quite interesting, if you're eating a kilo of white rice, that's a lot, actually. A dry rice for a kilo, if you actually heat it up with water, it's probably 2 kilos. I mean, if you try to force yourself to eat 2 kilos of rice a day, it's a disaster, I think. Normally, when I originally come from China, as you can probably tell by now. So when I was in high school, I eat about 400 grams white rice per day. But now, actually, I changed my diet because I'm moving to Australia for nearly 12 years. I eat probably only around 50 to 100 grams. It's quite interesting when you look into this curve, when they actually did this trial. You can see the trend. The more rice you eat, there's probably more relative risk you will have type 2 diabetes. But when you look into this line, you can see there's someone eating 400 grams rice. There's almost no risk for them to develop type 2 diabetes. I mean, for people actually doing this clinical trial, they know, because the subject is different. And, because people are different to each other. That's probably the reason. And then some people, you can also see here the same, someone eating rice, I actually get less possibility to develop diabetes. What's happening there? So one thing is the people are different to each other. Second is rice are different to each other. In all these trials, they use all different rice. So which actually indication is when you're eating rice, it not necessarily give you type 2 diabetes. It really depends on what rice you eat. And typically, and people were thinking, you probably heard lots of this story in the last 10 years saying GI, white rice have very high GI. What's GI is actually called a glycemic index. So it's around this area under this curve. After 2 hours, you consume a glucose, 100 gram glucose or 100 gram of white rice. And they reference those numbers. So typically, people tend to believe actually, if you eat a white rice, And within the first hour or two, you will have a very sharp peak of blood glucose rise in the first hour. And then, but if you're eating other low GI food, they will give you a more smooth curve out. But the recent research in the last 5 years, people realize actually they can't actually follow this called GI or glucose response. They should really follow on the insulin response, because when you actually have this high blood sugar, first thing your body will produce a lot of insulin, try to actually bring those sugar out of your system. The reason that you have a type 2 diabetes is because every time you have this high blood sugar and the insulin kick in, and then you keep using it, and your cell become not that sensitive. So the insulin index, I think it should be actually used more often than the GI, but not many people agree with it still. And when you actually look into whether how to find the rice actually can have this low GI in terms of give us a lower risk for type 2 diabetes, you will first thing people will look into is starch, because nearly more than 90% of the stuff in rice is starch, and then less than 10% is protein, and less than 1% is lipid. Which is quite interesting, because most of research on starch, because we come from a small university, we can't compete with the other large universities like UQ. As I know, lots of scientists work on the starch, and also, so we actually start to actually look into the protein and the lipid, which is have a proportional, have lyse actually, proportional lyse in the rice, but they may have more importance in terms of these their quality. So what actually people found quite a while ago is when they actually run the rice, a different rice through the clinical trial, on the insulin response, you can see some give you a really sharp insulin response, some give you a bit mild insulin response. And quite interestingly, these three rice they use is quite common. A rice variety, so they all have a 26% of amylose content, and 26% 26 or 27% amylose content, because in the literature, you probably heard of this story. If you eat basmati rice, you got low GI because the amylose, they got high amylose content. But when these rice all have similar amylose content, they still can give you a different GI and different insulin index. Which indication, rather than the starch and amylose, there's something else also play a very important role. And this is actually even earlier research when we actually reviewed the literature. What we found is, if you're eating lots of carbohydrate, it certainly give you higher, glucose, blood glucose sharp peaks. But if you're eating this carbohydrate with some fat together, it gives you a very mild peak on the blood glucose response. But for insulin, it doesn't actually reduce the insulin response as they reduce the glucose response, which is quite interesting. So I think there's a lot of things we don't know. And that's actually give me some sort of confidence we should actually research the correlation between starch and lipid. And so I did my PhD through Greenfield CRC on wheat before. So we found when we try to find this sort of low GI compound or something, because I was trained as a phytochemist, so we did the bioassay and the fractionation, and accidentally we found this phospholipid in it, and which have some sort of glucosidase inhibitory activity. And also when we actually try to extract it, we found it extremely hard to get it off the these succinate extraction. So. And then because we got a group of people actually research rice in Southern Cross University, so we sort of form a group and try to actually dig it deeper in the rice. What we found is quite interesting is we wrote a review. So for me, is I often want to actually write a literature review and publish it before I actually dig it deeper. So what we found is in rice, the lipid is actually present in two basic form. One is in the endosperm, and the other side is in the ambule and also the alveolar layer. In the alveolar layer, it's quite common. Is in other oil seed, you have this oil droplet there, And then in endosome, it's actually quite unique for cereal. So the lipid actually complex with the amylose in the endosome, the starch in the endosome. So this is a typical helix structure of amylose. And some lipid actually in the endosome actually complex with this helix structure, which is quite interesting. So I didn't expect it. So I suspect it's because some lipid complex with the starch. That's why when I doing research on wheat, I often have this sort of incomplete extraction of the lipid. And so during the research, we found there's so many of these phospholipids in rice, and then you can see they all have this fatty acid, phosphate, and also these different groups like a choline group or ethanoamine groups there. And in the review, we also found this phospholipid and lipid actually, in general, have very importance for the rice quality first. And this lipid will get rancid, and so it's, with the rice storage, it's very important. And also this phospholipid and actually, basically this lipid actually affect the physiochemical property, which is actually if you have the lipid in the starch, the geolatin temperature will rise as well. And also you have these, if you have this starch lipid complex, it basically reduce the digestibility of, it's not reduce the digestibility, the starch will be still digested, but the starch will be digested slower than before. And so in that way, I think because it's slow down the digestion speed of the rice starch, it basically can maybe actually provide some health benefit. But when you look into this detail in the literature, and you found actually there's the whole phospholipid about synthetic pathway, everyone already know it. But there's still no one actually know where this particular lipid called lysophospholipid, how they actually complex with starch and how they get in there in the starch granule, no one know, which I think could be a very interesting story and to go with. But you were thinking why people haven't done this before, and during the research we found it's just so hard. Based on the previous method, it's so hard. So the traditional research of cereal lipid, what they did is they passed the lipid in the silica column and then do two-dimensional TLC and then cut this block out and do derivatization and then put on the GC or GCMS to have a look, see what they are. This whole process takes a lot of time and also costs a lot of money. So, and the sample throughput is too small, and also it's because it's hard to it's kind of really hard to reproduce the result as well when you go through this. And then you can find, because the recent development of technology, you found there's a lot of LCMS, MS there, which people can actually research in most actually in human or animal science. So they, for example, they can research this phospholipid in human plasma, also liver, and they can use this and they can easily pull out all these phospholipid and compare it, which is quite fantastic. So I was thinking maybe we can use this method to do some lipid analysis for the cereal. What we did is we went in there very carefully to try to replicate the research. And so we compare all the extraction method, compare all the grinding method, and compare all the extraction method, and then try to simplify it, and then publish a paper early this year. We basically simplify this method to a very short extraction method, and then we can run around 70 samples a day, which is when you have a population of a mutated rice variety, we can go through 70 samples within a day, and we can easily screen through a few thousand within probably a month or two for your whole population. And so that's the purpose to actually develop this method. And during the method, we found, for example, this is one of the chromatogram you will see from the LCMS. We use a single eye monitor, and so you can actually see each of the lysophospholipid in this rice. And this method is quite sensitive as well. We can go easily down to sub ppb level. And then each analysis taking around about 15 minutes, so it's very quick. We correlate the data together with the apparent amylose, which is quite interesting. There's not much, people already know there's not much actually this lysophospholipid in the called waxy rice also is a low amylose rice. And there's some correlation there, but if you're not including this point, there's very much no relation between the apparent amylose content. So we did some collaboration actually with a Chinese university, Chinese university called Zhejiang University. So they give us, they provide us some rice sample, which is have a great represent actually geographically, over world. And when we screen them, we found there's a great difference between the total lysophospholipid content, and we also test every single of lysophospholipid in them as well. And the quite interesting story is that they all have a quite different profile. And what we found is actually all these differences mainly caused by the genotypic variance, not from the environment, because we actually grow the rice in two seasons, and we didn't find much difference was caused by the environment. And so we try to actually do some cluster analysis and PCA analysis. Basically, through this analysis, we can group them together, but it doesn't make sense. We can't actually group them into the previous subspecies level. And so there's a lot of things we don't know what actually control the biosynthesis of these phospholipid. And so we also did some correlation analysis to see whether there's a correlation between the amount of different lysophospholipid. And what we can see is for the phospholipid have a similar fatty acid. It doesn't matter if have ethanol, amine, or choline group. They have a very good correlation here. You can see. So this is kind of give us some idea how these phospholipid been biosynthesized. So I also worked with another researcher called Terry Rose. So he is an agronomist. So he interested in actually these the phytates in rice, which is another major phosphorus component in cereal. So we want to see whether there's a correlation between phytate and a phospholipid during the grain development. So we actually grow this rice and try actually tag all these panicles with different colors and follow it for 30 days of the grain development, and to test in every single time point how much phospholipid there, how much phytate there, how much total phosphorus there, how much free phosphorus there. And so we get a quite interesting story there as well. So you can see this is the whole grain weight. So it's increasing through these after the antithesis is increasing, which we all know that, expect that. And this phospholipid also increase almost in a very similar trend to that. And there's also other things we're trying to correlate together, which is quite interesting. I'm trying to publish this soon as well. And I guess I think just try to conclude is so my talk is I think there's a lot of factors to control the GI of rice. It's like a cooking, variety, and all sort of things, starch there as well. But lipid is a very small component in rice, but I think it may actually play a bigger role than 1%. So that's what I'm trying to say. Okay, and just finally, just acknowledgement. And so Graham is my boss, and Dan, and Terry is the agronomist. And so we also did some collaboration with Professor Jin Song Bao, and Chuan is his student. So Chuan actually came over to Australia, and we did some experiment together. And we also thankful this Australian China Science Research Fund. So give us opportunity to go to China and to visit all other researchers in China as well. Thank you.